problem. No problem. This is. Sorry, I didn't bring my pen. No problem. But but this is frozen also. Why is this undoing?
Cada letra con un cuento diferente.
You want to stop it?
Muy artesanal.
Have to stop?
Está hecho todo a mano.
Do it again. You just drag it.
Entonces cada palabra hay que ponerla en su sitio, Sí, porque si la enfoco automáticamente se salen más.
Vaya puta mierda, ¿qué es eso? Tanto peor. No lo veo, 800 para 3 slots, cuando al final son 3 slots.
No, pero son 3 slots, pero es que hay que hacerlo aparte, entonces hay que hacerlo software. Esto hay que romperlo más el recuadro,
No, that's crazy. That's completely crazy. Okay, 1 minute. I will. Yeah.
Rahul's computer also broke down.
Okay. Yeah. Okay. We'll just do this. Okay. This guy. We move it here. Yes.
El name.
This when you. Okay, so you just do this one. I will do this one.
Oye, si a las 3 empezamos podemos ir. ¿Cómo era eso en la universidad?
En 10 minutos, ¿no? 15 minutos.
Todo el mundo decía que empieza, no, pero era mentira. Todos esperando al profesor, cuando llegan a llegar y saltamos. Hay una norma que a los 3 minutos que llega el profesor te puede ir.
Todas mis vas a ver nuestros que también
What is your name? Yeah? You can now understand.
Yo creo que no.
Pero ¿qué universidad habéis ido vosotros? Nunca has ido a Serviva si querías.
No, la cosa es que si el profesor no venía aquí en esta persona, ¿cuánto tiempo tenías que esperar en la clase? ¿Toda la hora?
A ver, tío, es libre y la clase no.
No, que la cosa es que te pusieron falta o no. O sea, si no ha venido,
Ya, pero si el profesor ya queda media hora y él quiere dar la clase, es que si el profesor lleva más de 10 minutos antes, en teoría, pero en la teoría se haga la Pero si tiene libertad de cátedra, entonces puede hacer lo que quiera, porque tiene libertad de cátedra.
la tu cátedra.
Yes. Okay, fine. Yeah. Joshua, exactly. Is this on or?
Okay, sorry about that. Things happen from time to time. But anyway, I'm very glad to introduce our speaker today, which is Professor Rahul Pandit from the Indian Institute of Science in Bangalore. Rahul got the first master in physics in the Indian Institute of Technology, a second master in physics in the University of Illinois at Urbana-Champaign, where he also got his PhD back in 1982. And after some postdoctoral period in Cornell University, he moved in 1984 to the Indian Institute of Science, where presently he is the chair of the Division of Physical and Mathematical Science, with 99 faculty and 314 PhD students. In 2004, he organized StatFis 22 in Bangalore, and since there he was involved in the StatFis Commission, C3, of the International Union of Pure and Applied Physics, where now he's presently the chair of this commission. Rahul is very well known for his work on phase transitions, nonlinear dynamics, and turbulence. Current interests, to my knowledge, include the problems of turbulence, Ganglion and Navier-Stokes turbulence in particular, problems of superfluidity and Gross-Beta-Sp or nonlinear equations, and also the topic of his talk today. So it is a real pleasure to have you here. Welcome.
Well, thank you very much. Thank you, all of you, and especially Maxi for having me here. And my profound apologies for this problem with the things. You know, man proposes, computer disposes. So, all right, I'll make do with what I have, and the movies, I'll try to hold up my laptop so we can work with that. All right, so today I'll talk about cardiac arrhythmias. We do study many problems which have turbulence in them, in fluids and so on. But today I will tell you about that aspect of our work which looks at electrical wave turbulence in cardiac tissue. And I will try to tell you why that's important. This has been done over several years with several students who have now become colleagues, and several current students, and Sasha Panfilov, who's at the University of Ghent in Belgium. We have had generous support from various funding agencies, and we have published many papers which I will not bore you with, but I will try to give you a concise overview of some of the problems that we have studied in this area. We got into this area because of sort of some theoretical reasons. We were studying spatio-temporal chaos in extended dynamical systems, and then we found that the equations we were looking at were very similar to the simplest ones that are used to model spiral wave turbulence in cardiac tissue. So then we said, okay, let's look at it there. And then we got ensnared because the biologists don't want simple models. So to have the biologists take you seriously, we had to look at their complicated models. And I will give you an introduction to that. I will give you an introduction to mathematical models for cardiac tissue. Then effects of inhomogeneities in cardiac tissue, and to things which probably, if you're a typical physicist like me, you don't understand these words, and I will try to explain to you what they mean, and so on. And then we will also look at some examples of suppressing cardiac chaos and why that's important. So we will do this based on very detailed numerical simulations. I will explain them as we go along. But first, let me tell you why this problem is worth studying. First of all, cardiac arrhythmias like ventricular fibrillation are a major cause of death in industrialized countries. Typically, 1 out of every 6 deaths is because of ventricular fibrillation. All of you have probably seen this in the Hollywood version of the movies. You know, patient falls down, and then the doctor comes with these defibrillators and gives a shock, and all is well. So at some level, this gives you the mathematical basis. This talk will introduce you to the mathematical basis for all that. You will see that this ventricular fibrillation is associated with broken spiral or scroll waves in cardiac tissue. And the overall goal is to understand the dynamics of such ventricular fibrillation in the presence of tissue heterogeneities and so on, and to develop low amplitude defibrillation techniques. So all of you must remember this from your high school. That's the human heart. Left atrium, right ventricle, left ventricle. At some level, this is the most important because this is the ventricle that is responsible for pumping oxygenated blood to all parts of the human body. And then equally important is the electrical circuitry here. That's the sinoatrial node, which is the natural pacemaker of the heart, which sends these waves out, and it comes through some complicated set of connections through all these things. And then there are these pathways called the bundle of His, and there's an atrioventricular node, and then there is these other pathways which have to do with Purkinje fibers, which conduct electrical signals rapidly, which we will get to later. This you can find on the internet on various places. At a practical level, the heart is one of the most efficient electromechanical devices. It beats at about 72 beats per minute, typically 2.5 billion beats in an average human lifetime. And the rhythm is maintained by the electrical activity in this heart muscle. But these arrhythmias, ventricular tachycardia or ventricular fibrillation, destroy this rhythm. And here is a trace from some patient. Let's call the patient A. Here all was well. Now you can strap a machine and take a Holter test and do it over a long period of time. All was well. That's the normal ECG. Here, even to the untrained eye, something is happening. This is a fast heartbeat, and this tachycardia is, roughly speaking, Latin for fast heartbeat. And over here, even to the untrained eye, you see it's even worse. It's not only a fast heartbeat, but it is sort of very disordered, at least in time. Okay? And this is, if you like, ventricular tachycardia. This is ventricular fibrillation, which is life-threatening. In the absence of medical intervention, from here to death is 2.5 minutes. That's all the time you have to act. And it is associated with the formation of spiral or scroll waves. Here you see one visualized on a canine heart. Normally small mammals don't get such things. Their hearts are too small, so then it can be induced artificially, as must have been done in this research experiment. But on that scale, unfortunately, humans are large mammals, so humans do get such ventricular fibrillation. So here's an animation which I can't show you there, but let me show it to you here. Perhaps this is a small enough room that you can see it. This is the normal plane wave. This is a single spiral wave. That's a broken spiral wave. And those of you who are close enough can see, oops, it's fine, that there are measurements in real cardiac tissue which also show such waves. So this is not pure fiction based on someone's mathematical model, but is actually linked to experiments of three sorts. Three important types of studies provide our knowledge of this field. One is in vivo measurements, in vitro measurements, where you lay out some cardiac cells on a petri dish or some sort of slide. And then equally important, in silico studies on computers, which are now able to look at very serious models. So excitability, in a strict mathematical sense, cardiac tissue is excitable. I will tell, that means that there is a threshold. The membrane potential of cardiac cell must exceed some -60 millivolts before something called an action potential is observed. I will tell you what that is in a minute. And once excited, a cardiac cell cannot be excited anymore during a refractory period, which is of order 200 milliseconds. So here is a cardiac action potential. That's millivolts on the vertical axis, time here. And if you excite the cell, this is the resting membrane potential, -84 or so millivolts. It goes up quickly because of depolarization, fast inward flow of sodium ions, a plateau associated with slow inward calcium current, repolarization because of outward potassium current. All right, so what do the models look like? This is what we are trying to describe. The models typically are reaction diffusion equations of this form. They look very, very simple superficially. Delta V by delta T, V is the transmembrane potential, current density, capacitance density of cardiac tissue, and this is sort of different cells talking to each other through gap junctions. So this is a diffusive coupling. Strictly speaking, D is a tensor, but for the moment, let's just pretend it's a scalar. I'll tell you more as we go along. And then there are various models. I will show you two models, one of the simple sort, which is a two-variable model, and then one which begins to show you the complexity of the underlying biophysical system, and then more and more models. And there are different models for different mammals. I mean, there are guinea pig models, there are rat models, there are pig models, and more there are human models. For a while, when there weren't enough measurements, there were mixed models with some information from some mammals, some information from some other mammals. All right? So here is a simple model due to Panfilov, which shows the sort of spiral breakup that we were talking about. I'm sorry, on this slide, V, the transmembrane potential, is E. So here, this is of the general class of Fitzhugh-Nagumo type models. Let's not worry too much about that, but I will just, so here, this keeps track of how the membrane potential is evolving. All other information, which we will get to in a minute, having to do with ion channels, etc., is bedded into a slow variable. And again, this looks simple, but even at this level, actually, it is quite complicated because this function f of v is specified by certain inequalities, and there are many parameters, and these parameters are fixed by fixing to the typical spiral wave frequency that is seen in cardiac tissue, etc. If for those of you who do fluid dynamics, there's nothing as simple as the Navier-Stokes equation, okay, which we know is hard enough to solve. So we are solving now problems which are much, much more complicated. Okay, you can do it in many different ways, and believe me, we know how to do these types of simulations, so I won't bother you with simulational details. Here is a picture of a spiral wave turbulence in that simple model, all right? And you can see that the spirals are broken up. That is a pseudo-color plot of the transmembrane potential in that model. Red is high voltage, so that's where the action potential has gone up, and blue is the quiescent area. So let me show you this animation. I see you here. We have started a spiral wave, and you can see how it is evolving and how it is distorting. And if you see two waves collide, you will see that they don't pass through each other, but they annihilate. That's because the medium behind the wave is in a refractory state. It cannot be excited again. So when these waves collide, they don't go through each other. They just annihilate. Okay? And this is what we want to avoid. What I showed you is a mathematical analog of ventricular fibrillation. We want to get rid of it. But before I tell you how to get rid of it, let me tell you what a real model, which will get a biologist talking to you what that real model looks like. Okay, and this is not even the most sophisticated model around. They keep on changing every few years. All right, so this is biologically realistic. It incorporates details of ionic currents, ion channels, ion pumps, and ion exchangers. Okay, there are 12 ionic currents, 12 gating variables, intracellular sodium, ion, potassium concentration, calcium concentration in all these objects in the cardiac cell, which is known as the cardiac myocyte. Okay, so Maxi said make it a colloquium level talk, but don't leave out all equations. So with his permission, I'm putting up a few equations. Okay, so this looked deceptively simple. T and NP stands for Tentusha, Noble, Panfilov. Okay, those are the people who came up with this model. So it looks like this. All looks very simple until you say, what's that current? Well, that current is the total of all the ionic currents. Sodium, calcium of some sort. They're different types. They all got different names, so I won't explain. These are the exchangers which exchange these things, and pumps and so on. Then inward currents cause depolarization, outward currents cause repolarization. Then there is this Nernst potential which tells you when the electrical and chemical gradients are balanced. Okay. Then for each ion channel, you have basically something like an Ohm's law. Current is some conductance times the transmembrane potential minus that Nernst potential. Okay. But actually, the ion channel might not be completely open. So actually, here is the probability of the channel being open. Okay. And each one of these gates have some variable epsilon associated with them. Each one of these gating variables obey differential equations of this sort. Alpha and beta are the rates at which these gates open and close. Okay. And then you can work out, so there are time constants. And then these alphas and betas depend in turn on the voltage in some horrible nonlinear way, which I will not bring myself to write in a colloquium. It takes many pages, and they depends on the voltage, and they have things which, if you look with a physicist's mind, you get completely confused because their units are different. They use units in a way which electrophysiologists use. Eventually you understand it. So I'm not writing the rest of these equations, which take many pages. Okay. When you have these equations done and you're a good graduate student, you know how to solve these things, you can do it. All right. And you use Neumann boundary conditions to specify the problem completely. Otherwise, it's not specified because the atria and the ventricles are insulated from each other. Okay. Eventually, you have to go to a big machine. You have to do parallel computing. You have to worry about domain decomposition. And you have to make sure you get linear speed up, so people let you onto supercomputers. And my students certainly know how to do that. And then you can ask, how do you actually get the same spiral waves in these models? And the answer is yes. So I'm showing you one such in this, in an even more recent model than the one that I told you about. And you can see that with different parameter values, you either get no breakup or single spiral wave or something which breaks up. This is one single rotating spiral. You get another phase where something seems to start breaking up, and then it breaks up completely. For the level of this talk, I will just stay with these pseudo color plots of the transmembrane potential. But you can analyze time series, you can do power spectra, you can do all the things that we learn about in nonlinear dynamics to analyze these signals. And just to show you what it looks like, I'm going to show you some animation. And you will see three different parameter values evolving. And the first one will go to a single spiral wave, which is the mathematical analog of ventricular tachycardia. The middle one will be some sort of meandering. And the third one will eventually be that broken spiral turbulence state, which you see on the right-hand side. So you see it's broken up. Okay?
Finally, to really tachycardia. So the normal thing would be plane waves.
Yeah, normal thing would be just plane wave going through it and the heart beating nicely because of the signal that is coming from the sinoatrial node. But this, and you see when it breaks up like this, if you look at it under some conditions, you will see that the cardiac, the heart is just quivering like this. And when it's quivering like that, it becomes incapable of pumping blood. And if it becomes incapable of pumping blood, then the brain gets starved of blood, and okay, patient is no more in 2.5 minutes. All right, so the multi-billion-dollar, literally multi-billion-dollar industry of defibrillation. Okay, if you're lucky enough and a medicine acts fast enough, you administer some medication. But typically you use electrical means, and this is what you see in the Hollywood movies. There are two paddles, and you put it on the chest of the patient. It's a large electrical shock. They normally quote it in joules, but if I convert it, it looks like 5 kilovolts to me. And so it's a big shock. But it can save the patient on good days. And in high-risk patients, they have this implantable cardiac defibrillator, which looks like this. You can find them on the internet, and they're implanted sort of like this in some cases. So there are some catheters that monitor the well-being of the heart. And if it looks like things are going bad, zap, they give a shock, which is less than the one with the external ones, but still apparently feels like being kicked in the chest by a bull. But on good days, it saves the life. But there is a need to go for low amplitude defibrillation schemes. And one of the points of this talk is to see whether we can use our spatiotemporal understanding of this dynamics to see if we can design low amplitude defibrillation schemes. Okay, so we actually came up with a simple way of, with a low amplitude defibrillation scheme. You remember I told you that when two waves go through each other, they don't propagate beyond because it's refractory beyond that. So we said if we excite the tissue on some sort of a square mesh and we pass current along the edges of that mesh, then it becomes refractory, the spiral wave does not go through, and if it doesn't go through, then it gets absorbed by the boundaries. I mean, basically because each small domain is like it's too small for it to sustain spiral waves, and they get absorbed, and the spiral wave goes away, right? So let me show you some pictures. Basically what we have done, this is with no control, you don't see any things. Here you can see the lines. These are the lines along which we are exciting, and within less than 1,000 milliseconds, the spiral wave is gone. Okay? So I will show you a movie later, perhaps, to show you. I'll show you all the movies at one go towards the end, so that I don't keep on interrupting. Now you can ask, oh, but that's on a small domain. Will it happen on a bigger domain? That's the first thing we check. The answer is yes. Even on a bigger domain with a slightly more involved mesh, I mean a more dense mesh, we can do that. Does it work with a realistic model? I mean, that was a baby sort of Panfilov model, 2 variable. Probably it was, you were lucky you could do it. Turns out it works even with a more complicated model, which has got all the ionic realism in it. Now, the really hard question is 3 dimensions. Now, you're putting a 2-dimensional grid. If you did the 3-dimensional analog, you would have to cut into the cardiac tissue. You don't want to do that. So the question is, can you just put a mesh on one side of the heart and then somehow hope that it propagates into the 3rd dimension and gets rid of the wave? Now that didn't work as well as we would have liked it to. We got it to work for sort of slab domains where it's not too thick in this direction, and there also we had to use some cleverness where we had to pace it. But again, I don't know of anyone who's done better. So the full 3D problem, I think, remains a challenge. But it was good enough that at least the government of India granted us a patent. These are my students at that time. Having got the patent, a poor physicist can feel happy. But of course, industry is much more conservative than a poor physicist. We talk to various things, and they say, oh, until you've done animal trials, we won't even talk to you. So, okay, this will take a while. Okay. Now, you might ask, you see, I mean, so far I have been pretending that cardiac tissue is homogeneous. Actually, it is not. There are, all sorts of inhomogeneities in there. For example, there can be scar tissue left over from some previous arrhythmia, which leaves scar tissue, which is non-conducting. So that will be like a non-conducting inhomogeneity. And there were lots of experiments out in the experimental literature saying that such inhomogeneities lead to more spiral breakup, that is ventricular fibrillation in our mathematical analog. Partial suppression, that is ventricular fibrillation, became ventricular tachycardia, and complete suppression of ventricular fibrillation. So there's a bewildering array of results and contradictory, so we didn't understand them. I will show you how some of these experiments are done. Some are done, I think, if I'm not mistaken, this is chicken cardiac tissue, and all they've done is punched a hole through it. So that's the inhomogeneity. That is obviously non-conducting. But other, these are the in vitro examples I gave you. You place these cardiac myocytes on a glass slide, and then you can leave a hole or something, and that will be the inhomogeneity, right? So we introduced several types of inhomogeneities in our simulations, and the first thing we realized after a lot of systematic work was that sometimes these inhomogeneities did cause spiral breakup, so that was like ventricular fibrillation. In some cases they suppressed it partially, and in some cases they even suppressed it completely. This was what the experiments were finding. And what we were able to systematize is that this happens because of very sensitive dependence of the spatiotemporal evolution of these spiral waves on the position of this inhomogeneity. Okay? So for example, in this position, when we place the inhomogeneity, which is a square here, the spiral wave just eventually went out of the domain and was absorbed by the Neumann boundary conditions at the boundaries. In this position, the spiral wave gets anchored to the inhomogeneity and goes round and round the inhomogeneity, which is like a single spiral wave. So that's the mathematical analog of ventricular tachycardia. And in this position, excuse me, I said something incorrect here. This doesn't go away, it stays here, so it is still a spiral wave which is broken. So that's ventricular fibrillation, that's tachycardia, single rotating spiral, and this one, it has gone away completely. And the only thing that has changed in these three simulations is the position of the inhomogeneity. So to paraphrase Lorentz, sensitive dependence on initial conditions. Here you have sensitive dependence on the position of the inhomogeneity. Okay? And this is not restricted, this is not restricted to 2D. We will see that it also works in 3D. So here I have a little drawing based on our simulations that if I place the inhomogeneity where the red squares are, then the system will continue to show spiral turbulence. If I place it where the blue squares are, then I will get single spiral that is anchored to the object, that is like ventricular tachycardia. If I place it on the green squares, then I will get no spiral at all. And the sensitivity of this dependence shows up. If you try to blow it up, the boundary between these regions seems to be fractal, which seems to be the spatial analog, I won't give you every detail, of the fractal basin boundary between different domains of attraction in a high-dimensional dynamical system. Now you might say, does this work in 3D with an obstacle? The short answer is yes. This is for a realistic model in 3D, and here is the cylindrical inhomogeneity, and believe me, I'm not taking you through every detail, that the qualitative result is exactly the same. Depending on the position of this inhomogeneity, you can get all three possible behaviors and so on. This you cannot see very well, perhaps to amuse you at the end of this talk when, yes, sir.
When the spiral web start, do they start from the same place in the heart, or they can start?
Okay, so thank you for asking that question. If you actually took one of these models and naively put it in the computer, I guarantee you will not see a spiral wave. You have to work hard to get a spiral wave. One of the standard ways of starting a spiral wave is the following. It's called the S1, S2 perpendicular wave protocol. You start a spiral wave from the bottom edge, you pace, then a plane wave starts growing through the medium. It's a nonlinear wave, but it's a plane wave. Now after some time, you start another wave from this end called the S2 wave. And these two waves collide and they have a point, and then a spiral grows around it. So you have to work hard to do it, but you can do it. And there are analogs which go through in 3 dimensions to give you a scroll wave. A scroll wave, to a first approximation, is a spiral continued into the 3rd dimension. Okay? So you have to do that to get it started. Right? I mean, you can't see very well in this one, but let me see if I can show you at least one movie to show you how. Okay, now this is an anatomically realistic simulation domain. You're talking to biologists, you have to do something anatomically realistic. Where does this thing come from? Thanks to diffusion tensor magnetic resonance imaging. You have, for some mammals at least, on certain websites, the full coordinates of the left ventricle, the right ventricle, the septum separating them, and even more, the muscle fiber orientation. So your simple diffusion scalar has become a full diffusion tensor, and if you work hard enough, you can put in everything, and we have. Okay? So I'm going to show you a broken scroll wave in an anatomically realistic simulation domain. I don't know whether you can see it very well, but that is anatomically realistic. This is certainly what they do not teach you this when they teach you mathematical physics. When you solve Poisson and Laplace with nice boundary conditions. Okay? It's still going on, and that's, I think that is probably broken, and so on. And that's the goal. I mean, you really have to control that in the anatomically realistic thing, and you're still talking of a mathematical patient, not even a real patient. Okay? All right, so there are many things you can do. I won't show you. There is sensitive dependence on how many obstacles, how you do. You can get trapped on 2 obstacles. Those of you who are interested, I can show you later after the talk. Now, conduction inhomogeneities are not the only inhomogeneities that happen. If you look at the details, I told you there are these different ion channels. There are many parameters that describe ion channels, but amongst most important are the conductances of these channels. In fact, many of these medicines that are invented by these pharma companies are such that they modify the conductances of a given channel. So you have these calcium channel blockers, you have all these sort of blockers that are being prescribed to people all over the world these days. But sometimes, because of some condition or some damage caused by an earlier infarction, sometimes you get ionic inhomogeneities. That is, in some region, the conductances of some of the channels have become different. Okay? So can we study those? And the answer is yes. And you can. And so here I'm showing you simulations with a conduction inhomogeneity. You can study it at the level of simple models, or you can study it in realistic models where you actually have a handle on the conductances. In essence, the results are similar, that there is sensitive dependence on where you place the inhomogeneity. The one qualitative difference is that the wave, the core of the spiral, can actually go into the region of the inhomogeneity, because it's not completely non-conducting, it's just that it's got different conductances. So, well, it goes in, but, and it can, have very interesting behaviors, like it can be sort of chaotic inside, but not chaotic outside, and so on. But those details I will not worry you with today. You can do it in 3D if you're a good student and enough computer time. I mean, now we're talking serious amounts of computer time. Okay, then you can ask, does this control scheme that I was advertising, does it work with these inhomogeneities? The short answer again is yes, and I won't show you all the details. So you can see that mesh which I told you about, and that works with these obstacles, with any amount of ionic realism that you want to put into the model, with all the different ion channels that you would like to put in. It turns out that's not the end of inhomogeneities. There are other cells in cardiac tissue. The cells we have been talking about so far are the normal cardiomyocytes, which are excitable in the sense that I defined excitability. Okay? But there are other cells called fibroblasts, in particular cardiac fibroblasts. They occur often in the process of wound healing. So, for example, if somebody has some sort of infarction, after that, these cells multiply, They also grow as people age. And experiments, at least most people agree that they're not excitable, although some people have models of excitable fibroblasts. And they can basically be modeled at the level at which I'm talking about as RC circuits. And then there are, of course, different values of coupling parameters, which I will tell you about, but only in brief. So here are different stained slides from biology experiments. The first two are for sheep ventricular myocardium. The second are from the rabbit ventricular myocardium. The cells labeled F are fibroblasts. The cells labeled M are myocytes. Here again M and F, M and F, and M and F. And you can see that they are really interwoven, and strictly speaking, everybody doesn't agree on how fibroblasts are distributed in cardiac tissue, probably in different mammals or different patients, the distribution is different, and so on. So there is some need to study in computer models what are the possibilities, what different types of models could we have, and what effects do these fibroblasts have. So this is the myocyte equation that we saw already. There's a similar one here for the fibroblasts, except that the fibroblast equations are such that it's not excitable in a way that I can make precise as we go along. So we have studied different types of models for fibroblasts in our group. In one class of models, we have envisaged the bilayer model. So we have sort of myocytes sitting here and fibroblasts sitting here, and they are coupled through some fibroblast myocyte coupling. It also turns out that fibroblasts are much smaller than myocytes, so we also allow more than one fibroblast to couple to one myocyte. And in the simplest models, you don't let the fibroblasts communicate with each other, but you can also have models where they communicate with each other directly. Or you can have a monolayer of myocyte cells M, and then you have a random distribution of fibroblasts in that medium. So many of us in many different settings in physics study the propagation of waves through random media. Well, here we have, if you like, the propagation of a nonlinear complicated wave of excitation through cardiac tissue, but which is now also random because of the presence of fibroblasts. The question is, what happens to those waves as you have these models? So here is the bilayer model I told you about. The gray cells are the fibroblasts, the black are the myocytes, and this sort of tries to show you the different types of couplings we might have. And here is the random model that I told you about, where the red cells are myocytes and these blue ones are fibroblasts, which are distributed sort of like in percolation, site percolation theory, okay, of fiber. And then you have these equations sitting on top of all this, all right? These are numbers for the conductances to be chosen in a region which is biophysically reasonable. And then you can ask, what do these things do? Suppose I have a homogeneous myocyte fibroblast composite. Is it going to change the way in which my spiral wave evolves? And well, the answer is yes. You can have, you see the spiral tip, it will start meandering, so it is becoming a little closer to spiral breakup. Or it can break up completely into two spirals. Here we are in a region where it has broken up into different spirals, and you can analyze it in great detail using methods of nonlinear dynamics, which I would get into. You can ask what happens if you have a fibroblast inhomogeneity. I mean, the fibroblasts might not be distributed homogeneously through the medium. So just like we study conduction inhomogeneities, we can study fibroblast inhomogeneities. And we can find regions of parameters where they behave like conduction inhomogeneities. Let me remind you, in the case of a conduction inhomogeneity, the spiral wave does not enter into the region of the inhomogeneity, which is what is happening here. But with some other parameter region, they can actually enter into the region of the inhomogeneity as they did when we had ionic inhomogeneities. Okay? And then you can have randomly distributed fibroblasts. That is in the model that I told you about, where we have a random distribution of fibroblasts inside the medium, and you can get very rich behaviors, including no propagation at all. And so let me show you one animation perhaps of this. So you have different parameter regions, and you see the granulation in the waves now caused by the randomness of the domain, okay? And sometimes it breaks up, or sometimes they will just stop propagating, like in the bottom one here, the spiral wave is just gone. There is too much inhomogeneity. I mean, I don't want to call it localization, I mean, whatever, but absence of diffusion in a random medium, okay? But of a very, very complicated nonlinear wave. You can do it in 3D too, which I'm not going to show you. So here is a cartoon that as we increase the number of fibroblasts, you go from a single rotating spiral to no spiral at all. SA stands for spirals absorbed by the boundary. And you can do that in 3D too. And at a reasonable concentration of fibroblasts, all wave propagation is gone. All right, does the control work when we have fibroblasts? Again, short answer is yes, our control scheme works. You can then say, so far, I have been pretending that there is no mechanics here, but all of us know that our heart beats. And why was I pretending that? Because at the first level of modeling, the assumption is that the mechanical system is enslaved to the electrical system. So if I understand electrical waves, the mechanics will follow suit. But in more advanced models, people are now beginning to look at mechanical deformation. We used a very simple model, which is not fully all right, where you sort of let the diffusion constants change depending on how much you have stretched the medium, either in the x direction or the y direction, or both. But actually more advanced models do full finite element analysis, and also they introduce a new piece of physics which is called extension induced currents. Okay, so even the how much is going through the ion channels depends on the local extension of that part of the thing. So I won't give you the details, but okay, all these things do change the behavior of spiral waves in the system. But the good news is, at least at the level of 2 dimensions, our control scheme works. I just want to show you that once you have mastered the techniques required to run such programs on the computer, you can keep on adding more and more complicated cells. You will remember in the first slides, the first few slides that I had, where I had this heart, I showed you there were some special cells called Purkinje fibers, which are very good at conducting rapidly. Now, nobody quite agrees, or nobody knows enough exactly how the Purkinje fibers are distributed. Some people say it's a fractal, some people do real measurements on a rabbit and say we will believe results only if you put in exactly that real network. We also took the point of view that it is advantageous to look at simple models first, where you connect one layer of endocardial standard myocytes with another layer of Purkinje fibers and connect them either at every site or at every other site and so on, and see how the spiral wave dynamics evolves. And you have to write down complicated sets of equations, which you can. So here is our bilayer sort of model. You have myocytes here and the Purkinje things here. And again, I won't give you every detail, but we can study how the spiral waves get affected when you couple them to a layer of Purkinje cells. Let me see if I can show you one quick simulation. Oops, forget it. All right. So, and here, I don't know how much you can see here, we have a sort of ramified network of Purkinje fibers, and we see how it is the signal is going from the Purkinje sheet to the other sheet. So let me skip all that, and you can do it. Now, I didn't show you a real example of scroll wave breakup, so I will show it to you in a minute. But first thing, I hope I've convinced you that at least at a mathematical level, we can begin to write down models thanks to many, many measurements. By the way, these measurements have been going on for centuries. The earliest measurements probably go back to a person called Mines, who was in the University of Montreal, if I'm not mistaken. And he actually even was found dead in his lab one day, if I'm not wrong, probably because some of these experiments, probably he got an electrical shock himself while doing some experiments and so on. So the long history of this studying these spiral waves and it becoming clear that there were spiral waves involved. And so I think most of the people in this trade would believe now that ventricular fibrillation is associated with the breakup of spiral or scroll waves induced by this sort of, in the jargon of the trade, reentrant activity, because it comes back into the system. Spiral turbulence is spatio-temporally chaotic or turbulent in whatever manner you understand turbulence. And these durations of these chaotic transients depend on the size, and we have checked this in many simulations. Small size, no spiral waves eventually. Large size, lots of. By the way, those of you who haven't seen this YouTube, you should go and look at it. Just Google a whale heart. Finally, they got a huge whale heart. It's big. Was it 80 kilograms or something? Okay. All right. So, and spiral breakup in these models can be controlled by the sort of low amplitude pulses that I told you about. Our simulations show that these arrhythmias depend sensitively on the shape, size, position, etc. of these models, which sort of, at least to me, looks somewhat bleak. People talk of personalized medicine. If it has to be personalized at this level, I don't know, but okay, all right. And then some of our work has shed some light on the sensitivity and provided some natural way of understanding the wide variety of experimental results. And we can study many types of inhomogeneities. So let me thank you for your attention. But let me show you one last movie to show you that we keep on working. And this is a movie of a scroll wave in an anatomically realistic domain for a human heart. And if you watch carefully enough, you will see the scroll wave breaking. And in this particular case, it is breaking because of a gradient in the fibroblast density. I mean, in fact, some gradients are found from the lower point, the so-called apex, to the top. So sometimes they're helping, but if it's too much of a gradient, it can cause scroll waves to break. And I thank you for your attention and for your understanding for my.
Unfortunately, that we could not see all the movies from Smith, but I think we've got a general picture. Now we have comments or questions. Single.
Yes.
It's really fascinating to see what level of detail you can really model the dynamics of the situations in the heart. Coming back to the original motivation, the defibrillation.
Yes.
Seeing all these variety of phenomena and heterogeneity and individual phenomena, do you see a chance that one can get away from a sufficiently globally valid?
Well, I am hoping yes, because you see, one of the reasons was that every time I introduced some new complication, I also showed you that at the two-dimensional level, the scheme was working well. Now, so the challenge is not these complications, the challenge is the third dimension. So if we can solve that third dimensional thing in any model, I'd be willing to bet it will work with all the complications.
So for a medical application would not be sufficient to worsen my 5%.
You're right. Well, I mean, there's always a default option, which is already in our defibrillators for those who are, it depends on your point of view. I mean, they're fortunate enough to have the internally differently implanted ones, but yes. But we were hoping for low amplitude defibrillation. Yes.
Yes, so the third dimension is really important because the spinal is let's say, system size, not as large as the system size. So why is the third dimension so important?
Oh, because, I mean, it's actually a scroll wave, right? You're right, it's tiny. I mean, if you look at the dimension of the heart, it's absolutely right. The dimension of the cardiac wall, it is much smaller, but unfortunately it is still too large. We've done the sums. Even with the realistic third dimension, at least our pulsing scheme doesn't go all the way. I don't know of anyone who can control reliably in the third.
The problem is that you want to add surface and doesn't enter.
Yes, exactly, absolutely, right.
The spiral can propagate
Yes,
below.
I mean, of course, if I put a 3D grid, I can do it, but that's like cutting into the heart. There might be some new methods having to do with optogenetics. I mean, all of you know about optogenetics? Okay, you've heard it in the neural context. So people do it in the heart, and whether that can help us go into the third dimension, I don't know. I don't think anyone has shown it in a believable way so far. Okay.
More questions? Comments?
So the vehicles like the whale heart, so those are people more easily get fibrillation. Is that a fact that the whales get more?
Well, okay, I don't know, is the honest answer. I even tried to get first a somewhat smaller mammal, namely an elephant, and I discovered it's even hard to get an ECG for an elephant, partly because the elephant doesn't, cooperate, and the skin is very thick. Okay, so you see, it might even be more complicated than that, because formally I say it's a bigger heart, but all of us like to work in dimension-less units. So one suggested way, which we have suggested, Panfilov has suggested, is to say you scale the thing by the wavelength of the wave in that medium. So that requires measuring all the proper conductances and so on for the whale heart. I don't think such studies have been done. Possible. I don't know. I mean, I just don't know the answer to whatever I mean, nobody has written down a complete equation for the set of equations for whale heart.
Okay. Any further question, comment? If not, thanks again, Rahul.
Thank you very much.